Hi, I'm Karen Collins, and I'm here today with Professor Ellen Crozier from the School of Medicine at the University of Glasgow in the UK. He's a world-renowned expert in the study of compounds in plant foods that you might have heard about called flavonoids. You may think of them as antioxidants, but that's part of what he's going to talk to us about today. You've been in this field an awful long time, you've seen a lot. What frustrates you most that you hear people saying that you think they have clearly gotten mixed up or gotten the wrong message?
Well, there's a lot of information or papers in the literature, even in quite eminent journals like PNAS, of cells of, say, that are subject to prostate cancer, or neurons from the brain in culture. There's one example of neurons in the brain being treated with blueberry juice actually, and they were protective. But the neurons in your brain, if you drank bilberry juice, because of the metabolism within the body, would never, that's modified compounds in low dose which reach these cells. So putting bilberry juice on prostate cells, any other juice in fact, or tea on prostate cells, neurons, any other in vitro cell system does not provide evidence of protection, but it's easy science. And also, there should now be a realization that in reality things are a bit more complicated. You have to test the compounds which enter the circulatory system, which are modified from the compounds that you ingest.
And the hard part is, in doing these more advanced studies, is also that actually we're not all the same because of they
Yes.
come from our digestive processes.
Yes.
So it's a complicated thing to get that research
A lot
right,
of what goes on with things we digest eventually ends up in the colon, where compounds are broken down by the colonic microflora. Some of the things in raspberries, for instance, called elagitannins, break down to ellagic acid, which is converted to compounds called urolithins. And the ability to break down the big elagitannins to simple ellagic acids varies enormously from one person to another because of their colonic microflora, which is a reflection of their genetics, what they've eaten over a long time, and how they're feeling, and time of year, and all sorts of things. So that after you've eaten raspberries, the amount of urolithins which are produced can be 0 to 8%, depending on individual volunteers. So particularly when it gets to the colon, we're all different.
So you and I could eat the same amount of these plant foods and have completely produced different things.
We could produce different levels of these
and
potential...
therefore benefit differently.
Yes, exactly.
So sometimes when we see results in studies, the results might be not statistically significant because there's so wide variation,
Yes.
which old school would mean, so it's probably could be chance.
Yes,
but new school is, it could also be that
new school,
some people it matters and some people it doesn't.
New school means more replicates, so you can demonstrate it's significant.
Good. So one of the interesting experiments you were talking about today was some work you did with, was it mice or rats? Hamsters. And looking at fatty streaks, which is one of the very early stages in the development of atherosclerosis, or what some people used to call hardening of the arteries. And you looked at several different foods and had some interesting findings.
Yes. Well, firstly, our model, because with humans we all have a different lifestyle, and for the majority of people, or with some it develops earlier, we don't get heart disease, or the symptoms of heart disease don't appear till we're 50, 60 or 70 years of age. So doing experiments with humans to look at protective effects is very long term, very expensive. And there's a model of hamsters who do develop hardening of the arteries, fatty streaks in their circulatory system, the same way that we do. They live for about 2 years normally. You can put them on a high-fat diet, and within 3 months they develop clear evidence of fatty streak deposits on their arteries. 20% of the artery wall can be covered with fatty streaks. As well as putting them on a fatty diet, we gave animals raspberry juice, green tea, black tea, bilberry juice, a whole series of juices and teas, thinking we could get a very nice league table of what was protective and what was less protective. And in fact, they were all amazingly protective. given over this 3-month period at doses which are equivalent to you and I having a couple of glasses a day. So it's a realistic intake, and there was clear evidence of protective effects in an animal system which mimics the way we're likely to operate, but much more rapidly.
Mm-hmm. So it's a fair representation.
Yes, yes.
Okay, and I think in the talk you said it's equivalent to about 300 mils.
Yes, about
So
300.
about 10 ounces of juice, that would
Yes,
be.
yeah.
That's pretty exciting.
Yes.